Hello students, welcome to Padhobio. My name is Shashikant Chaube and in this video we will discuss about cell membrane, also known as plasma membrane. So as of now we know very well that cell membrane is very crucial for a cell because it maintains a boundary between the cytoplasm of the cell and the extracellular environment. So let's start from the first and discuss the composition of the cell membrane. Cell membrane contain a variety of biological components like lipid, proteins and carbohydrate as well. Its structure was given by different scientists, but the structure that resembles cell membrane most is the fluid mosaic model, and this model was given by Singer and Nicholson. This structure shows that the plasma membrane contains lipid, proteins and carbohydrates, and this model also represents how these structures are available in the cell membrane. Composition of cell membrane is not fixed, but it changes continuously to maintain its fluidity and also as per the requirement of environment. This lipid bilayer provides a basic fluid structure to the cell, and at the same time it provides a impermeable barrier to most of the water soluble components. Also, the cell membrane consists of three classes of amphipathic lipids, that is phospholipid, glycolipid and sterols. Amount of particular lipid depend on the type of cell for which we are talking about, but phospholipids are present in most abundant amount. The lipid molecule is known as amphipathic. Do you know why? Because it has two ends. One is hydrophilic or the polar end, and other one is the hydrophobic or the non-polar end. Main phospholipid in the animal cells are the phospholipids, having a glycerol backbone and a fatty acid chain, in which two long chain fatty acids are attached through ester bond with the adjacent carbon atom of the glycerol backbone, and third carbon atom is attached to the phosphate group, and this phosphate group is attached to the several other head groups which may be ethanolamine, serine, choline and etc. This makes its phosphatidyl-ethanolamine, phosphatidyl-choline and phosphatidyl-serine. And another class of phospholipid which is important to know is sphingolipid. Here the backbone is sphingosine rather than glycerol. Sphingosine is a long acyl chain with one amino group and two head group at another end. So we are discussing this because like glycerol lipids, sphingolipids are also present in the cell membrane in a major portion. For example, sphingomyelin is a type of sphingolipid that is present in the myelin sheath of neuron. Now talking about another component, that is carbohydrate. Cell membrane also contain carbohydrates mainly in the form of glycoproteins but also some glycolipids. So as the name suggests, glyco. Here glyco means glycan or oligosaccharides which are linked to either lipids or proteins. Here glycoproteins, it means proteins are attached with oligosaccharides through a covalent bond, and this bond formation takes place through a process known as glycosylation. This glycosylation may be either N-linked glycosylation or O-linked glycosylation. Though there are many types of glycoproteins present in the cell, in which most common are N-linked and O-linked glycosylation. Later we will study this topic in detail in further videos. But today you need to know, during N-linked glycosylation, the oligosaccharides are linked to the nitrogen atom of the protein, and during O-linked glycosylation the sugars are attached to the oxygen atom of the protein. Now coming to the topic, carbohydrates are present on the cell surface because they play an important role in cell-cell recognition, where they come in contact with any cell and share some information. Now coming to the last and another important component present in the cell membrane is protein. So as you can see in the fluid mosaic model, that proteins are present in three different forms: peripheral proteins, integral proteins, and the last is the lipid anchored proteins. They all have their different functions. For example, peripheral proteins are present in the membrane either associated with integral membrane proteins or at the peripheral region itself. They are present at the interior as well as exterior of the cell membrane because they tend to have very loose connection with the cell membrane. Once reacted, the molecule get dissociated from the membrane and go inside the cytoplasm to activate another component. Example of these protein includes phospholipase C, cholesterol oxidase, and cytochrome C. Now coming to integral membrane proteins. They span the membrane bilayer and have a cytosolic domain which interacts with another integral component present inside the cytosol, and that's why they are known as transmembrane proteins. The hydrophobic domain of these type of protein consists of either alpha helix or beta sheets or both as well. Example of integral membrane protein includes G protein coupled receptor, ion channels, and proton pumps. Now the last one is lipid anchored protein. These proteins are also attached to the surface of lipid, but they are very tightly associated with the lipid molecules using covalent bond. So mainly there are three types of lipid anchored proteins available, that is prenylated lipid anchored proteins, acylated lipid anchored proteins, and glycosyl phosphatidyl inositol linked lipid anchored proteins, that is GPI proteins. Prenylated proteins are the proteins in which different proteins are attached with different isoprene units at specifically cysteine residue. Here there is a motif present in the protein, that is CAAX. In this motif, C is the cysteine, AA represent any aliphatic amino acid, and X represent the type of prenylation that is going to be occur. So here majorly three types of prenylation is seen: farnesyl, geranyl, and isopenoid groups are mainly seen to be attached. Prenylated proteins play important role in cell differentiation, cell morphology, and cell growth. So coming to another type of lipid anchored protein, that is acylated proteins. Here different fatty acid chains are incorporated with amino acids and result in acylated proteins. So most common are myristic and palmitic fatty acid chains. And the last is GPI linked anchored proteins. Here the proteins are linked with GPI complex via amide linkage at the C-terminal carboxy group. But great diversity was seen in the GPI linked anchored proteins, due to which major functions are possible, that is cell adhesion, cell receptors, and some hydrolytic enzymes. So that's all for the cell membrane. Hope you like this type of videos, and if you like this type of videos, just go and subscribe padhobio channel, and see you later in the next video.